Good morning, everyone. It's a pleasure to be here today on behalf of my team and of Michael Taylor, who is our fearless leader. So the last talk was actually very moving, and I could see some people tearing up in the audience. You're going to be tear-free for this presentation because I'm going to talk about the research that we do and from a perspective of sitting down all day in front of a computer to try to get at answers for these brain tumors. And so you might want to have another sip of coffee instead. So I'm going to tell you a little bit today about pediatric brain tumor called medulloblastoma. And really, in the last few years, we've come a long way in understanding such tumors because of genomics, which is a field where we study mutations or errors in the DNA that occur in these tumor cells. And so I'm going to give you a little bit of background on medulloblastoma today. I'm going to talk a bit about the primary disease and then what happens when tumors recur. And just a brief overview of our insights into how we might do better at treating some of these tumors. And so medulloblastoma arises in the cerebellum, this is the back of the head, in about 1 in 100,000 children. And these are actually treated quite successfully with a combination of surgery, chemotherapy, and radiation. And so we have survival rates of 85%, which is really excellent compared to some other brain tumors. And even for higher risk patients, we have survival rates of 70%. High risk, typically, these patients are already at diagnosis. They have the spread of the disease onto other parts of the brain or the spine, and so they end up doing actually a lot worse. But even in the best case scenario where we can cure patients, there are significant long-term side effects, and as you can see here. And so we'd really like more tools in our arsenal for treatment so that we can do better for these patients that we can cure, and so that we can actually cure some of the recurrent and metastatic patients where we just can't really make headway. We can treat only about 5% of them successfully. And so one of the ways we classify tumors currently is using a grading system from the World Health Organization, which is based on histology. So this is looking through a light microscope at how the cells look in the tumor. And this is good in some respects, but actually one of the major drawbacks is it doesn't give us any insight into what's happening inside these cells. A lot of them look the same, but have different outcomes. And so I'll just give you one example of that. These are two sick kids patients where almost everything was the same except their outcome. And so you can see they're both 6 years old, both girls. They had successful surgeries. They had the same chemo and radiation treatment. And so their tumors look pretty much undistinguishable under the microscope. And yet the first patient is still alive after 5 years, and the second succumbed to metastatic disease after just 3 months. And so what could be causing the differences in these outcomes? And it turns out that genetics can actually give us a pretty decent answer. And so in the last few years, we've performed a lot of genetic analyses on medulloblastoma, and now we know that instead of one disease, medullo patients actually fall into 4 main groups that have distinguishing demographics, they have different clinical features, and they have different errors in the DNA, so they have different mutations. And so these tumors, while they look the same under the microscope, actually behave quite differently. And that first group, these patients actually have an excellent prognosis. They never recur, they never metastasize. Sonic hedgehog tumors have activation of a signaling pathway called sonic hedgehog, that's why they're called that. And those tumors tend to come back in the same spot they were resected from. And group 3 and 4 tumors are the ones we know least about, and those tend to metastasize to distal sites, and their prognosis is a lot worse. And so when we consider these two patients in light of their genomic information, we can see why their outcomes are so different. One is a Wnt tumor, and she does very well, and the other is one of the group 3 tumors with a very dismal outcome. And so knowing the subgroup of tumors can help us to better characterize patients and think of these groups differently, but it's also actually a really essential piece of information we can use for design of clinical trials. And so here I'm showing you two examples of two agents that are in clinical trials. And if we didn't have subgroup information, we might have failed these agents because they actually end up having only 3 out of 20 patients who respond. But those 3 patients are 3 out of the 5 sonic hedgehog subgroup patients. And so we know that instead of failing such drugs, we should actually conclude that they're excellent for a specific subset of patients. So it's very important to subgroup the patients. Now, just a note on clinical trials. Most clinical trials actually recruit patients who have already recurred. So their primary disease has been treated, and they either have metastatic or recurrent disease. And I want to emphasize how important it is that we actually know something about the recurrent disease and how it is different than the primary. Because unfortunately, a lot of clinical trials for targeted agents, so for mutations, end up failing. And so what the current paradigm is to biopsy or analyze the primary tumors, and then once that patient recurs, so they failed therapy, to apply targeted therapeutics using targets found in the primary, but to the recurrent tumor. And we think one of the reasons this strategy fails is because the recurrent tumor is actually really genetically dissimilar. So it's very different than the primary. And so the assumption was that that's not the case. And so we tested this hypothesis, and we found that that's exactly what happens. Treatment induces many, many changes in the cells that make up a tumor. And we published this work earlier this year. And I just want to show you one example from that paper that makes this point really well. And so this is the case of a sonic hedgehog medulloblastoma patient. And you can see here going up, this is the cellular content of the tumor. And across the bottom, we see time. And so at the beginning, this patient's tumor was very small, and we can see how the mass grows with time until surgery. And pretty much when we look at the genome of these cells, we see that they're patch1 mutant. That's one of the classic mutations in this subgroup of patients. And yet after this patient recurs, after more time goes on, and we take a second sample from the relapse, we find no evidence of patch1. Instead, the tumor is now made of a related cell that coexisted before the patch1 mutation occurred. And now that it had the opportunity, perhaps it also had a mutation that made it resistant to therapy. So now it expanded, and the second tumor is actually made of this completely different genotype. And so you can see how important it is that we know what we're actually testing drugs on in these clinical trials, because this patient would have failed, for instance, a smooth inhibitor because they don't have the patch1 mutation anymore. And so this prompted us to look more closely at these primary tumors and really get at understanding how uniform or different they are. And so we always assume that all the cells are pretty uniform, but we can see that they behave differently. And now that we know what to look for, we can see evidence of those cells that ended up making the secondary in the primary tumors. And so we undertook a study to kind of rate these tumors, medulloblastomas, on a scale of heterogeneity, where on one end we have genetically uniform or homogeneous tumors. So it doesn't matter which cells you take, what biopsy you take from a patient, you're always going to have the same answer. These are the mutations of interest. All the way to the other end, where we have genetically heterogeneous tumors, where if you take different biopsies, you may have a different answer as to what mutations are present. And so we know that this is clinically relevant because in many other cancers, which I've listed here, we can see a difference in survival for those tumors that are uniform, which are shown in blue, and those tumors that have lots and lots of different colors or clonal lineages. And so you can think of these genetically diverse tumors as just having a lot of genetic opportunity for becoming resistant. So they just have this repertoire of possibilities that you don't find in a uniform tumor. And so our study design was basically to look at 36 tumors. These are medullos, some high-grade gliomas, another brain tumor from adult as opposed to pediatric patients, and some renal cell carcinomas. And from all these tumors, we had between 4 and 11 pieces, which is really remarkable. And we asked how informative is just one biopsy versus multiple biopsies. One of the most important pieces of information we can know from medulloblastoma is subgroup. So is subgroup the same across a tumor? That was one of our main questions. And also, are targetable or actionable mutations similarly present throughout a tumor, or do we need to biopsy multiple times in order to finally collect all the information we might need? And so for the subgroup question, we were very happy to find that it doesn't matter which piece of a tumor, which biopsy, here shown by little circles. It doesn't matter where you profile from, subgroup is always the same, it's stable. And so we were very encouraged by this finding. A Wnt tumor is always a Wnt, a group 3 tumor is always a group 3. It's not the case for GBM, this is something we knew before. So in a GBM, it depends where you biopsy, you may find a different answer. And so it's much more clinically difficult to make a decision based on subgroup in GBM. But for medulloblastoma, we know that's all we need. For the genetic component, so are mutations uniformly present in a tumor, we applied sophisticated algorithms to discern the genotypes of cells. And so here I'm showing you each cell is a little hexagon, and the color basically corresponds to a genotype. So pink cells may have some mutations, green cells have a slightly different set of mutations, and so on. And so depending where you take your biopsy, you may have different constituent proportions of these different genotypes. And what we can do is build a phylogenetic tree, which is shown here on the right, that basically cluster things by similarity. So all the pink lineages end up together on the same branch, green stuff ends up together, blue stuff ends up together, even though they come from different biopsies. And so that's what we see when we look at patient data. So these are some of the trees we've built of the genotypes of cells from a medulloblastoma tumor in this case. And you can see that some of these long branches, for instance, is biopsy 5, it just has so many mutations that are just found in that biopsy and none of the other biopsies. For instance, KDM1A, which is a gene we know is important in cancer. And biopsy 6 down here doesn't share KDM1A or any of those other mutations. Instead, it has a TERT amplification, which is another really important gene in cancer. And so we can't treat these, we can't assume that they're the same because they're clearly very different. And they have mutations in genes that we know are relevant to targeted therapeutics, for instance. So in this high-grade glioma, we see that some biopsies, this little bunch of branches at the top, have an EGFR mutation. And so we could, if we biopsied this patient and took some of those cells, we would think they would be a great candidate for EGFR inhibitors. But it turns out that if we were to put a patient on such an inhibitor, they would not respond, or they may have an initial response and their tumor would come back because the rest of the tumor is lacking that mutation. And so there's heterogeneity. And similarly with this renal cell carcinoma, this little branch here has a mutation in P53, which we know is one of the main culprits for radiation resistance. And so the tumor may be eradicated, just leaving behind these few cells that could seed a recurrent tumor. So in clinical reality, no one's ever going to biopsy between 4 and 11 pieces of someone's tumor and do sophisticated genomic analyses. But we can actually do quite a lot with just 2 biopsies. And one of the things we can do is grade tumors on the scale of genetic heterogeneity. So we can tell, for instance, which tumors are these ones that we should worry about, which are the heterogeneous ones that present a treatment challenge, because we don't expect targeted treatments to work very well. But even for these tumors, we know already that there's something uniform about them. We know subgroup is uniform. And so while genetic mutations may be heterogeneous, other aspects about these different biopsies may be the same. And so that gave us the idea to consider immune-therapies. And so we've already heard about how the immune system can heal us, and that's actually really true. T cells catch a lot of cancers before they develop, but in case we need some help, there are immune therapies where we can train T cells to recognize a specific molecular flag that's present on the surface of cells. So as long as we can find a flag that's present throughout one of these genetically heterogeneous tumors, as long as we can find something that's uniformly present, immune therapies would actually be a feasible strategy for these tumors that are currently therapeutic challenge. And so just very briefly, I'll tell you that what I'm talking about are actually chimeric antigen receptor T cells, CAR T cells. These have been in the news for quite a while now. Basically, a patient's own T cells, own killer T cells, are taken out and re-engineered to recognize that molecular flag that we know covers cancer cells. And then they're put back in the patient, where they reintegrate into the immune system, circulate through the patient's body, and eradicate the tumor when they home in on it. And so we looked at a number of these receptors already in clinical trials, and so we looked to see if these look promising for our medullos that we know are genetically heterogeneous. And you can see 4 of them here, and I don't have time to describe these graphs, unfortunately, but basically all the little green dots are the different biopsies of each tumor, and you can see that they're very tightly clustered. And so these targets are molecularly uniform. And that is the case whether we consider the genetically heterogeneous tumors or the genetically homogeneous tumors. And so we think that this is a really promising strategy for getting at these really difficult to treat tumors that may fail in targeted therapy trials. And so hopefully I've convinced you that there's a lot of value in doing genetics and genomics on pediatric brain tumors and cancer in general. We can do more precise risk stratification using such information. We now know that there's a big difference between primary and relapse tumors, and that we should rebiopsy at relapse before enrolling patients on clinical trials. And we know that there are additional therapies we might consider that would work really well for these cases that would otherwise have failed. And so we still have a lot of work to do, but we're optimistic that our work will make an impact into patient lives and their families. Thank you.